Good morning. My name is Anna. I'm from Bohumolets Institute of Physiology in Ukraine. And first of all, I'd like to thank the organizers of the conference for giving me the opportunity to be here and to present the result of my works. I'd like to start with a short introduction. Hypoxia is one of the main causes of the optic nerve disorders that lead to gradual and often irreversible disruption of the visual signal transmission from the retina to the visual centers of the brain. Retinocollicular projections represent the first part of a secondary visual pathway, and its lesions could lead to navigation, orientation, and visual attention deficits. Here we studied effects of short-term hypoxia on retinocollicular synaptic transmission. In our experiments, the co-culture of dissociated retinal cells and superficial superior colliculus neurons was used as in vitro model of the retinocollicular pathway. The co-culture was prepared using originally designed chamber for co-cultivation. This two-compartment chamber consists of a silicon ring with a thin vertical glass baffle placed tightly on the cover slip. The main idea for creating this chamber was to spatially separate two suspensions at the earlier stage of cell adhesion to substrate. After the cell adhesion, the silicon ring can be removed and neurons are able to regenerate their axons and dendrites and restore their synaptic contacts. Here are microphotographs of the co-culture at certain day in vitro. The black arrows indicate retinal ganglion cells, white superficial superior colliculus neurons. Two-way arrows points boundary between cell population, and scale marker correspond to 100 microns. I'd like to describe the structure of the experiment. We use pair patch clamp technique in a whole cell mode and pharmacologically isolated NMDA, AMPA, and GABA-A postsynaptic currents were worked in superficial superior colliculus neurons by generation action potentials in presynaptic retinal ganglion cell. Method of fast local superfusion technique was used for fast and local application solutions needed on synaptically connected neurons. And the protocol of the experiment was as follows. Firstly, we applied control solution. This period we called control. Then we supplied for hypoxic solution, hypoxia, and then switched back to control solution, reoxygenation. And we observed that hypoxia up to 5 minutes induced long-term potentiation of NMDA neurotransmission, long-term depression of GABA-A neurotransmission, and temporary suppression of AMPA neurotransmission. Moreover, we observed that hypoxia induced a reduction of voltage-dependent magnesium blockade of NMDA response. Presence of magnesium ions in external solution at holding -70 induced their voltage-dependent blockade of evoked NMDA response in control. An application of hypoxic solution leads to the appearance of evoked NMDA response that disappeared again during reoxygenation. This reduction was not due to cell depolarization, as we can see it from the voltage-current relationships presented here. We also analyze hypoxia-induced effects on spontaneous and miniature activity. And here, as an example, I present the effects on NMDA activity. We observed that hypoxia increased frequency of spontaneous and miniature activity and also lead to a second mode appearance in amplitude histogram of the miniature events. These miniature events were recorded in nominally calcium-free solution with the presence of TTX. Under such condition, the release probability is minimal and fixed. So according to a binomial model, this appearance of the second mode can be explained by the increase of the average number of release sites. The estimates for binomial parameters also indicate such increase. And in conclusions, I'd like to say that all currents were analyzed in terms of a binomial model, and this analysis revealed that hypoxia acts mainly presynaptically on excitatory neurotransmission, and both pre- and postsynaptically on inhibitory neurotransmission. The results obtained reflect the electrophysiological basis of hypoxia-induced lesions of retinocollicular pathway. I'm sure about the importance of further research focused on identifying precise processes and mechanisms underlying such hypoxia-induced plasticity, and hope that they could benefit future treatment of pathological lesions triggered by hypoxia. Thank you very much for your attention.